In our lab, we are interested in understanding the regulation of apoptosis. Apoptosis is the best understood form of cell death, and it is critical for essential processes in biology. For example, it is very important during development, and here you have for the shaping of organs, and here you have a mouse paw, and you see this, the fingers are interconnected by cells that die during apoptosis, during development by apoptosis to give the individual fingers. The same happens in humans. Apoptosis is also very important for tissue homeostasis, and you can imagine that an adult organism has more or less the same number of cells. And this is maintained through a balance between cell proliferation and cell death. Now, you can imagine how when this balance is altered, because you have too little cell death or too much cell death, this can lead to disease. In the context of cancer, what you have is too much cell proliferation and too little cell death. And this leads to the overgrowth of tumors. And indeed, resisting cell death is considered a hallmark of cancer. Apoptosis is also very important for the correct functioning of the immune system. After an infection, you have a contraction of the T cells, and they die by apoptosis. Also, their selection during their development is also done by cell death, or cell death is very important for this process. And when you have alterations, then you can also, this can lead to autoimmunity or the loss of immunity. So one of the key steps in the regulation of the apoptotic pathway, and that will determine the point of no commitment, of no return in the cell's commitment to death, is the permeabilization of the mitochondrial outer membrane. And this is controlled by the proteins of the BCL2 family. This is a very complicated family of proteins, and they have, unfortunately, a very confusing set of names, because they are called BAX, BAC, POC, big, bad, BCL-XL. Forget about the names. The thing is that this family can be divided into three groups. The initiators, and these are usually BH3 only proteins that sense when the cell is under stress and will initiate apoptosis. Then we have the guardians. They are pro-survival. They inhibit apoptosis. And what they do is they block the initiators as well as the executioners, which is the third type. And these are the ones that will mediate directly the permeabilization of the mitochondrial outer membrane, like BAX and BAC and POC. But today I will focus mostly on BAX and BAC. You can see here a scheme of how they work together. So we would have these BH3 proteins that are initiators, and they are being sequestered by anti-apoptotic BCL2 proteins. For example, this is used in the clinics already. There was venetoclax is a drug that was developed to target these proteins. What it does, it blocks the sequestration of the initiators by the guardians, so that they are released and free to activate the executioners, activate BAX and BAC by binding to them, which triggers a conformational change and their oligomerization and insertion into the mitochondrial outer membrane. And now, once they are activated and they oligomerize, they induce the opening of a pore, the apoptotic pore, that releases the so-called apoptotic factors. And this includes some proteins that are usually in the intermembrane space, like cytochrome C or SMAC. Once they are in the cytosol, these proteins will induce the activation of caspases. And caspases are a family of proteases that cleave as a selected set of target proteins, so that when they are activated, the cell is disassembled in a very organized manner. Then there are these apoptotic bodies that are formed, and they are removed from the tissue by phagocytosis by the enabling cells or macrophages. So that when the anapoptotic cell dies, it is nicely and cleanly removed from the tissue, and nothing happened there. it's an immunosilent process. Now, recently it was recognized that during apoptosis, there is also the extrusion of the inner membrane and the permeabilization of the inner membrane through the apoptotic pore. And this leads to the release of mitochondrial contents like mitochondrial DNA, which are sensed as because of their resemblance with bacteria of bacterial origin. They are sensed as the presence of a pathogen in the cytosol. Actually, viruses or bacteria, if they expose their DNA to the cytosol, this will also activate intracellular immune sensors, in this case, the SIGA-STING pathway. And this induces inflammatory signaling and the production of type 1 interferons. So basically, what we have here is a process that would make apoptosis inflammatory. Now, as I told you before, apoptosis is happening, for example, in our immune system all the time, every day in our body, and we are not all the time inflamed. So most of the, usually apoptosis is immunosilent. And this is achieved because the caspases, when they are activated, they not only disassemble the cell, but they also cleave and inactivate inflammatory proteins of the inflammatory pathway. So they block the inflammation. This brings us to a situation in which when we have this mitochondrial permeabilization, two competing processes are triggered. On the one hand, the caspases that are going to be anti-inflammatory and try to have a very clean death process, and the extrusion and permeabilization of the inner membrane that is going to release mitochondrial DNA and initiate inflammatory signaling unless it is inhibited by the caspases. So we have this competition, and the outcome of this will determine whether apoptosis is indeed immunosilent or not. Now, I have this picture here. So this is how an apoptotic cell looks like in initial stages of apoptosis. And this is, I'm showing the executioner protein bags. Here is tagged with GFP, and you see that it is accumulating at these dots. We call them apoptotic foci. And I'll show you later, they are actually apoptotic pores. And you can see how they are not homogeneously distributed along the mitochondrial network, but assembling at specific points in the mitochondria. So in our lab, we have been trying to understand the mechanism how BACs and BAC and BOC mediate this permeabilization of the mitochondrial outer membrane. And we proposed a model in which these proteins, when they become activated, they form like a very irregular and undefined structures or pore structures. I say this because, well, their structure in the first place is not very heterogeneous. Indeed, we still do not have a structural model of these proteins in the membrane. There was one very nice paper this year, cryo-EM structure of PAX oligomers, but it still is in detergent in absence of a membrane. That gave some hints, but it's a very flexible structure, hard to determine or to solve. Now, this activated PAX forms dimers, and the dimers, we proposed that they form like a clamp at the edge of the pore that stabilizes the pore edge. Having an edge in a membrane is a very unstable process because it exposes it would expose the hydrophobic tails of the lipids. I mean, actually the membranes bend, I show you later, and this creates a line tension. So here what we think is that BACs and BAC, by forming this clamp at the edge of the pore, they are releasing or stabilizing the pore by decreasing the line tension. Now, these dimers form high order assemblies, and they do not have a defined stoichiometry. Now, if you think of the nuclear pore complex, beautiful pore in the nucleus of our cells, this is very well defined with a very nice architecture, a fixed number of proteins. Here we have a complete different situation. So we see stoichiometries going hundreds, tens, I mean a huge variation. And also when we look at these pores at the nanoscale, we see that their shape is also very variable. We see lines, arcs, and rings. And we could see with atomic force microscopy that both the arcs and the rings are associated with membrane pores. I'm gonna enter a bit more in detail in this. So for studying backs and back pores at the nanoscale, we use single molecule localization microscopy. This gives us an overview of a high resolution of the pores in an apoptotic cell. Of course, one needs to zoom in order to see these structures. I'm going to do some advertisement first. We were initially analyzing all of these nanostructures by hand. Very frustrating and time-consuming for the students. So we put some effort into developing a software. It's based on AI, and you can train it on your structures of interest from super resolution images, and then you can use the software to automatically detect the structures and perform quantitative analysis, as you can see here. You can classify them and then really do some quantitative analysis. Okay, now yes. Yes.
Dive on that a bit more, because I think that's like something super interesting. How can we? Okay, so question is like for AI-based quantitative analysis, this is like still an issue, right? So how we can leverage the power of AI without producing artifacts that are also hard to detect in AI-based platforms, so that we can actually do quantitative analysis. And I was just wondering if you want to elaborate a bit like how this actually works. I'd be very fascinated by that.
I would say, no surprise, the key here is the training. So in the training, the user has to train the AI with the images and the structures. So the user defines, in our case, we have lines, arcs, rings, dots, aggregates, right? So you need, in our images, to have a good statistics of finding or of automatic detection of the structures or classification. About 100. Okay. But I can imagine that this will depend on the type of the structure that you want to analyze, and your signals to noise ratio, the quality of the images. So the sensation with AI is always training.
so that really goes to this specific issue that I was thinking of, because let's say you have a bias in your training data towards certain structures, right? That you typically see now, but actually your true data that you have is like a structure that is not in the training database. Would that then be misclassified or something? So fully. How we can deal with that? Right?
I have to say this is the case, and but the human also does that. So in a way,
true.
In a way, when we had these images, we had a huge distribution of structures, very irregular. We wanted rings, we wanted structures like the nuclear pore, but no. We found a huge distribution, and we first had to figure out how to even analyze them, because that was the first, like, what do we do now with this? And then we made an arbitrary classification. So we ourselves defined what we thought could be the most representative structures. And then we left a huge like bag of unclassified things that are aggregates. So, which doesn't mean they are not, that they aren't anything, but we don't know what they are. So we can distinguish something as a ring or as a line or as an arc or something amorphous. But maybe the amorphous stuff is also interesting. We don't know.
Yeah, we're struggling. Absolutely, and I think we're struggling with similar things also.
Yes. I think, but this was the problem we even had, before this, because we did a lot of manual analysis in the sense of classifying and trying to the structures. So at the time when we decided to develop the software, we already had an idea of what we wanted. Now, if the user would have triangular structures or hexagonal structures, it could be trained into that. So the families or the type of structures that you want to look for in your sample are defined by the user. So it's arbitrary and defined by the user.
Okay, thanks.
Maybe this is something that you test on other structure than like any clustering or any other structure inside the cell, or is it?
So we have tried it for BACs, for BAC, for BOC, and for the LP1. In a way,
But not like completely like different structure, like focal adhesions or whatever. Like I was wondering how general your methods can be applied to different type of.
So I was once discussing with Melike Lacadamiali, I don't know if you know her. She was trying to apply it for filamentous structures that form larger assemblies, and she said it didn't work well. So I think in this case, the stuff we've used it for is generally discrete structures. We tried to apply it to other types of microscopy where we will have different type of images, and then it wasn't working as well as with super resolution. With Storm, it works very well. With STED, it works well. Even with AFM, it's not working to the same extent, and it's a matter of noise to signal to noise ratio. So these are the type of structures that we arbitrarily defined for our system. We found some dots and aggregates, and we don't really know what they are. Maybe they are still defined structures that are not in the right focus or the right angle of observation. But then we saw some structures that we've considered non-random assemblies. We classify them as lines, arcs, and rings. In some cases, we also saw some kind of double lines, but we didn't get many many statistics for this, so we left them out. We just put them as example, just in case someone would see them again. But as I was telling you before, one feature of these structures is that they do not have a well-defined shape or size. They really have very broad distributions, and you can see here for the rings, the radius goes between 20 to 50 nanometers for BACs. So rings from 40 to 100 nanometers in diameter. It's a huge variation. So my initial wish was to do averaging, not subtomogram, but averaging of the structures, and but it was impossible because the averaging worked very nicely for the nuclear pore complex. Now, we also applied this, as I told you, there are these two guardians, BACs and BAC, that have at that time thought it was thought that they had fully overlapping functions. And so we did the super resolution analysis for both proteins, and we found that they actually do form the same type of structures. But when we compare the sizes, we realized that BACs was forming larger and more heterogeneous assemblies than BAC, suggesting that the two proteins have a different way of assembling during apoptosis. Before I move into the problem of their assembly, we were very excited when we saw the rings because we thought, maybe this is the apoptotic pore. Nobody had seen the apoptotic pore before, so we were like, wow, look, now we can see it. But of course, seeing a ring in a 2D image doesn't mean that there is a hole. And this is, as I showed you before, we went back to atomic force microscopy, and this is in vitro in supported lipid bilayers, where we reconstitute BACs oligomers, or BAC, in this case, this is BACs. And then you can see how smooth a control membrane is, and this is in the presence of BACs. And you can see the formation of pores in the membranes, and the bright dots are protein density. And when we perform an analysis of these images, we were very happy to see that we could also define arcs, lines, arcs, and rings. And the distribution of sizes was very similar to what we had seen in the cells. But here now we could see that both the arcs and the rings, I mean, basically all of the rings and a fraction of the arcs were actually associated with membrane pores. Because with the atomic force, I didn't explain atomic force microscopy. I guess you know it. But let me know if any method you want an explanation of how it works. This gives us a 3D image of the topology, and then you can see here the hole in the membrane, right? So we were happy because actually this type of structures, at least the arcs and the rings, are associated with them. Then if we go back now, I was telling you we were very shocked by this different assembly of bags and back, that they were forming structures of different sizes. So we thought we wanted to see or to quantify their oligomerization in apoptotic cells over time. And for this, we used a method, or we implemented a method that we call single particle imaging by photon counting confocal microscopy. So basically here in this image, we take a set volume with several slices, and we analyze only the particles that are fully contained, their fluorescence is fully contained in the volume, not the ones that may be at the partially in and out. And then we detect these particles, and we calculate their intensity with a 2D Gaussian fitting. And now, from going from intensity or number of photons to a number of molecules, in this case, we need a reference sample. And the best behaving sample is, again, the nuclear pore complex. In this case, we used a protein called NAP96 that is well known to be a 32-mer. And we use basically the same imaging condition, the same tag, parallel to our apoptotic cells, so that we can measure the photons, the number of photons per particle in the nuclear pore complexes. And from that ratio, then we simply, with a very simple calculation, we can calculate back what is the number of particles or the number of fluorophores that we have per individual apoptotic pore or apoptotic focus. And here you can see some images of how the systems evolve during apoptosis. This is when at the time of mitochondrial permeabilization, so the very initial moment of apoptosis induction. And what you could see here, PAX is usually cytosolic. This I didn't explain before. It's usually cytosolic, and it accumulates into the mitochondria and forms these dots that you can see become brighter over time. These are the apoptotic foci. BAC is constitutively mitochondrial in healthy cells, and it is homogeneously distributed. But when we induce apoptosis, it reorganizes and assembles also into apoptotic foci. And we wanted to compare them, but to discard that whatever effect we may see was not because one protein is soluble and the other protein is mitochondrial, we also used a mutant of PAX that is constitutively mitochondrial. And when you activate this mutant, you can also see that it accumulates or reorganizes into these dots or apoptotic foci, right? And now we measure the stoichiometry of the particles of PAX and BAC, or the mutant of PAX, over time. You can see here a summary of the results, and this would be at 5 minutes, and then 15, 30. So you can see here the assembly of PAX and BAC. We decided for this, I think this way of showing the data is very good in the sense that you can visualize very clearly that it's a very broad distribution. I was telling you before, this is hundreds of molecular units. You can see the variation is huge. But when we pay attention, when we look at PAC, actually what we find is that the oligomerization is faster, but it's really not growing farther during apoptosis progression. I have to say we use in this case caspase inhibitors so that the cell is not being disassembled, and this extends our measuring time from 30 minutes to 60 minutes. And for the mutant BACs, what we see is very similar behavior to the wild type. So I think from these graphs, one can already conclude that BACs and BAC assemble in different ways, and that it is not their differential localization in the cell, but that defines these distinct assembly properties. Now here in this graph, we have a different way of looking at the same data. Now we are calculating, as if it was not single molecule, we calculate the average, and then we see how the average stoichiometry of the apoptotic pore grows over time during apoptosis progression. And you can see here for BACs, the wild type, you can have this linear growth, grows and grows and grows and grows. For BAC, as I mentioned, it seems to be faster, but then it saturates and stays more or less the same. And the GFP BACs behaves like the wild type in the sense that it continues to grow, 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 except that probably it grows faster because it's already at mitochondria. It doesn't need to find the mitochondrion. Now, so from these experiments, we concluded that the two proteins have different assembly properties. I'm not going to show you the data, but we did these experiments in cells that were double knockout, so that only one protein was being investigated, and these are the data, or single knockout, meaning that we would have the sister protein present endogenously in our cells. And what we found is that actually the BACs and BAC have these different assembly properties, but when they are together, they regulate each other oligomerization. And they do this because they co-assemble into the same supramolecular structures. And this we could visualize by using two-color STED imaging in living or dying cells. And you can see how BACs and BAC are co-assembled together into the same individual lines, arcs, and rings. So we thought, okay, how is this important, right? Does it matter for apoptosis progression in any way? You have to think, when I told you about what happens with mitochondria get permeabilized, one key step is the release of these apoptotic factors, cytochrome C and SMAC. They activate the caspases, but these are very small proteins. Cytochrome C is 12 kilodalton. It would need actually a very small pore to be released from mitochondria. So we thought that the structures, the large structures that we are investigating probably are not relevant for the release of cytochrome C. But mitochondrial DNA is large. So we thought, okay, maybe the dynamics of assembly are important for the release of mitochondrial DNA. And we now went back to a system in which we don't have GFP BACs or GFP BAC. We have simply wild-type cells or cells lacking BAC or cells lacking BACs. And then we measure the kinetics of mitochondrial DNA release during apoptosis. This we did staining the mitochondrial DNA and seeing how it colocalizes or not with the mitochondrial network. As you can see here, this is a healthy cell. And when you have apoptosis, you can see that the DNA is not colocalizing with the mitochondria so well anymore. So what we can see here, this is the time or the dynamics of mitochondrial DNA releasing in apoptotic cells in wild type. And what we see is that when we have BACs knockout cells, this means the pores are being made by BAC. The kinetics are basically the same. This was surprising because BAC makes the smaller structures. And initially we thought, okay, maybe the smaller ones will be not so good at releasing the DNA. No, actually they are pretty good. But when we don't have BACs in our cells, BAC in our cells means BACs is making the pore, then we see a delay in the release of the DNA, suggesting that it is not the size of the pore, but the growth kinetics that defines how fast the DNA is being released. And you say, Ana, doesn't matter the speed or the rate of the DNA release. I told you there's a competition with the caspases. So if the DNA is released very quickly, then it can induce STING activation maybe before the caspases are blocking signaling. If it takes very long, the opposite would happen. And indeed, I'm not going to show you here, but what we could see is that the levels of STING activation were higher when we had faster mitochondrial DNA release. Yes?
History dependence. So let's say like you have like a very quick but short peak. It's not big in magnitude, but it's super early. That would be more important than having more magnitude later on, something like that. Is that kind of like what you're saying, or? You say kinetic. So
yes, with the kinetics.
Versus, so let's say like speed versus magnitude.
Yes, we are saying that the faster growth of the pore will be more effective in releasing the DNA.
Yeah.
the magnitude will be important, but basically both pores, I mean, the smaller structures that BAC forms are also capable of releasing the DNA. The size is not defining the release. It's how fast it grows.
Okay, so it's not a trade-off, it's more like the kinetics is actually much more important than size.
Yes.
Okay,
Yeah.
I got you.
That's what I wanted to say. I was going to say something. Well, but basically from here, this would be a summary for this part. What we see is that the balance between BACs and BAC in a cell, the balance of molecules, will determine the growth rate of the apoptotic pore. And here I would like to highlight that the point, I mean, we're still calling it apoptotic pore because how people, I mean, I don't know how much you have heard about pores, but if you think of a pore or the concept of a pore out there is a little hole in the membrane defined by proteins, right? And those are just defined, not changing over time. What we have here already is a very different behavior. The apoptotic pore is actually growing over time, and the growth is regulated by the balance between BACs and BAC. And this is because they have different assembly properties, and we can find a functional consequence, at least, in the kinetics of the release of the mitochondrial DNA and the downstream inflammatory consequences. So for us, this was very interesting because it was changing our understanding of the apoptotic pore as a static, all-or-none thing into a dynamic process. Now, when the mitochondria get permeabilized, it starts with a very tiny thing and grows and grows and grows.
When you're imaging BACs and BAC, you're basically having a stationary image. So are you sure that, so to say, the BAC is not faster in opening and closing? So basically that pores open and then close, and then that when you look at the single time snapshot, that basically what you're capturing is like you have lower probability to capture a whole big pore if the process of opening and closing is faster.
Mm-hmm.
You understand?
Yes. We haven't done these experiments in cells, very difficult. But we have done similar experiments in vitro, in liposomes or in supported by layers. And actually, we never see the pore closing. So I think this is also a very important aspect of this pore. It's a pore that grows and doesn't close. At least beyond a certain size. But I'm going to touch into this. And looking at the time, I'm also going to skip some slides. Shashank's work to go now into this part is unpublished. But we got thinking like, okay, we see these lines, these arcs, these rings. How are they related to function? Do they all have the same function? I mean, for the line arcs and the rings, we can see they can be associated with pores. What about the lines? So we wanted to a bit to understand how the supramolecular assembly of these proteins would be correlating or associated or defining the permeabilization of mitochondria. And for this, we developed a correlative microscopy approach combining basically all of the microscopies that we had learned how to do with electron microscopy. So basically, remember I explained to you how we can calculate the number of fluorophores or the number of proteins in the individual pores. We can use super resolution microscopy to visualize this supramolecular organization. And now we want to correlate this information. So we correlated these two pieces of information or these two types of microscopy. And then we correlated further with electron microscopy to see how this is associated with the underlying mitochondrial ultrastructure. And well, I'm not going to go into the details of how this is done. I have to say, so far we were working on BACs and BAC overexpression. It's not that you can overexpress this protein much because they kill, so naturally we were working with low expressing cells. But now we generated endogenously tagged cells. And this is how this method would work. Basically, we get this is an image, super resolution image of our apoptotic cell. And now we can really correlate it with the EM so that you can see for now. If we zoom here, we see that we have like a almost closed arc or a ring, if you like, associated with a mitochondrion. And if you look there, you see that there is a pore in the outer membrane. Once we have this method, so here is another picture. You can see we can measure also then the size of the outer membrane opening and the size of the back assembly and compare them. And actually they are very similar in size. We can tell this actually contains about 17 molecules or 20, but more or less that order of magnitude, right? And then the EM is not cryo-EM, but it is a tomogram. So we can actually then construct a 3D image of the mitochondrial ultrastructure. And there you can see that there is a pore in the outer membrane and also a pore in the inner membrane. We were also very excited to see that. Okay, so now we have developed this method. We thought, let's now do some statistics and try to get numbers and see how these assemblies relate to function. So basically, this works. First, we take the photon counting imaging, and then we take the super resolution image so that we avoid photo bleaching when doing the stoichiometry, because the super resolution photo bleaches. But then once we have the super resolution image, we identify the structures that we are interested in, and then we find them in the stoichiometry image. And then we can calculate the number of particles or the number of fluorophores per particle. So now we can do some statistics and we can analyze the stoichiometry of the bulk particles when they are lines, arcs, and rings. And we were very disappointed because basically what we find is that the distribution of sizes is more or less the same. Meaning, like, well, what we could say from here is that the shape of the assemblies is not determined by the copy number, by the stoichiometry, right? And even you can see that if this is the length or the perimeter, depending whether it is a line or a ring, you can see that the length of the structure is increasing, like the rings are larger than the arcs that are larger than the lines, but the stoichiometry is more or less the same. So it's not really, it doesn't seem to be that the stoichiometry is relevant for the supramolecular assembly. Then we go now to the electron microscopy. We correlate the images, and I have to say here, what we find out is that the mitochondria are fantastic fiducials because they form a complex network that is different from cell to cell, and we can use the mitochondrial signal now to correlate the two images. And now you can see here how we have these back nanoassemblies, and they are associated with membrane pores. We find them very often at the tips of mitochondria. And then something that we also find quite often, and that was also very shocking to me, is this type of situations, and these are most often lines, where we see that there is a huge pore and the inner membrane has been extruded. This, I mean, you can see, I don't know if you can get to see here, this is a double bilayer and this is just one membrane. It's the same here, maybe here is clearer, right? So you really have the end of the outer membrane and then the inner membrane being extruded. And well, we did some statistics. I mean, in this method, the throughput is not super high, as you can see. But basically, as I was telling you, we see these pores happening mostly at the tips, and in some cases, they are in the middle of the mitochondrion, and they are associated with the extrusion of the membrane. Sorry? Well, it could be like a bleb. Actually, I want to see what you think about this that I'm going to show you next, because we were shocked by this, how often this extrusion would take place. Initially, I thought that would be rare events, but actually they are quite common. And in order to get more statistics, we went back to fluorescence microscopy, and now we did super resolution of the inner and the outer membrane and halo back. And there you can see that back is nicely at the edge of the apoptotic pore. And now I change, or maybe the name of pore to opening, because again, I was telling you it grows, but it grows huge. I mean, I don't want to call this a pore anymore. I think it's a different thing here. It's a huge opening that allows the release of the extrusion of the inner membrane. And back is at the edge of this outer membrane. And what we think is that the shape or the outer membrane edge is what defines the shape of the assembly that we see in the microscope, like the arcs or the rings or the lines are basically following the edge of the outer membrane in the context of this huge opening. That the membrane as an organizing principle for the oligomers. So we quantified this now, we can do better statistics, and what you can see here is that actually the extrusion process is quite common. And we were wondering whether this would be mediated by, or not mediated, but whether physical properties would be important in this process. And this is maybe here when I say that it looks like a bleb, but inside the cell. So our hypothesis here is that during apoptosis, I told you the inner membrane is permeabilized, and then because the ion gradients are disrupted, there is swelling of the mitochondrial matrix due to the entry of water molecules into the matrix, and this swelling would lead to the formation of this bleb, this extrusion. We were actually hypothesizing that this osmotic swelling would be a driving force in the extrusion of the inner membrane. And to test this hypothesis, we did a treatment from the 80s to the cells in which by adding malinomycin and igericin, one can trigger matrix swelling without changing the potential. And we did this treatment to our apoptotic cells, and then we quantified the extrusion, and we can see that when we promote swelling, we see an increase in the extrusion of the inner membrane, suggesting that this osmotic swelling contributes to the process. Now, coming from working with UVs in my life for many years, there was this nice paper from 20 years ago in which they could calculate, I mean, this is a giant vesicle with a lipid phase coexistence, a lipid order and a lipid disorder phase. And Tobias Baumgart and Amhes and Whatweb devised a method to calculate the line tension between the two lipid phases based on the geometry of the giant vesicles, because the line tension is going to try to minimize the perimeter, and by that it pushes the membrane out of the minimal energy configuration that would be a sphere. So you can use, we thought maybe we can use a similar approach to now calculate whether there is a line tension at the edge of the pore. Well, I think from the images one can already tell that there is a line tension, because otherwise the inner membrane would be adopting a spherical shape the same way that it is adopting or trying to adopt a spherical shape when it reaches the cytosol. And it is like the outer membrane is squeezing the inner membrane into this bell shape, right? So basically using the same approach now, we calculated what could be the line tension at the edge of the outer mitochondrial membrane, at the edge of the apoptotic pore. We got a value of around 7 piconewtons. That is very good, in very good agreement with the experiments that we had done some years ago with the proteins in vitro in supported by layers. So from here now we teamed up with Gerard Humer, and he simulated the process of extrusion. And in this movie, you are going to see in both the outer membrane is modeled as a rigid rod with a hole in it. And then the inner membrane is folded inside, and then the system is allowed to relax. And there's no mitochondrial inner membrane permeabilization. I'm going to start now the other movie in which there is the same situation, except that the mitochondrial inner membrane becomes permeabilized, and then there is osmotic swelling. And then what you can see Well, you don't need osmotic swelling for the for the extrusion. Here from this simulation, actually the unfolding of the crystal provides relaxation. I mean, the membrane becomes more relaxed, and this is sufficient for extrusion at a certain size of the pore. But when we have osmotic swelling, the process is enhanced. And you can see here also, actually this neck forming, and you can calculate a line tension at the contact between the outer membrane and the inner membrane. Very nice in agreement with our model. So based on this, we thought that then the extruded, or the mitochondria, where the apoptotic pore is related to an extruded inner membrane, would have larger pores because the inner membrane extrusion is kind of exerting a force on the apoptotic pore and making it larger. And then we measure this. We are now trying to get more numbers for this. This is only for the EM. We're doing it on the fluorescence microscopy, and we can see that actually this is the case. The pore is larger when it is accompanied by inner membrane extrusion. So from here, I summarize and finish. I went over time. What we found is that the back clusters colocalize with pores or openings at the mitochondrial outer membrane, and their shape is defined by the edge of the outer membrane opening. And what we find that there is this extrusion through the outer membrane, of the inner membrane through the outer membrane, that is promoted by osmotic swelling, but that it feeds back to the apoptotic pore in the outer membrane and promotes its growth. And from here, I think for us, something very shocking and interesting is that there is an interplay during apoptosis actually between the inner membrane and the outer membrane. In apoptosis, everybody was focused on the outer membrane, but actually it's both membranes, the permeabilization of both membranes that play together to govern the dynamic behavior of the apoptotic pore. And with this, I finish, and I would like to thank the group, the people who did the experiments, the funding agencies, and you for your attention.
Very much. Very fascinating talk. Yeah, we have still time for questions. So
Yeah, I would have two questions. One is basically just really for me to understand when you said like that the shape of the membrane defines the size of the pore. Do you mean that the proteins will co-localize with the local curvature?
We think so.
Have you done simulation? Because there are some, you can collaborate with people who are doing simulation. Maybe.
Well, Gerard is simulating that. I don't know the outcome. But and this is only possible now after this structure was published this year. That was our dream for a very long time. What we see in the AFM images is that the protein density is actually accumulating at the edge of the pore, and we also see this in the fluorescence images. With the AFM, we can measure the mechanical properties of the membrane by doing for spectroscopy, and what we see is that it actually reduces the line tension of the membrane when you pierce through it, having bags in them. We complicated the assay, but we could measure a decrease in the line tension of the AFM tip piercing through the membrane when we have bags in there. It's a very indirect way of measuring that this. So how we imagine them is that they are forming this type of structure at the edge of the pore, and that they stabilize the pore edge by decreasing the line tension. And by decreasing the line tension, then they also prolong the lifetime of the pore. And as you see, and maybe I didn't mention, we think that also this extrusion process defines the irreversibility of the pore, because once you have inner membrane extrusion, I cannot imagine how energetically the system can go backwards, right? So maybe before the extrusion, you can have smaller pores that can be removed. We don't know, if this would be possible, but I could imagine maybe. Once it is extruded, the fate of this mitochondrion is done, and maybe it can be removed by mitophagy, but otherwise there's no going back.
I have another question, which is more, so to say, conceptual. Have you at the very beginning, you say basically how cancer and other cell lines behave differently in apoptosis, but have you done all your experiment with basically cancer cell lines? Is there, I mean, have you thought about checking if apoptotic process is different in the, so to say, non-cancerous cell lines?
The apoptotic process is, I mean, the process, the things that happen are the same, but we were discussing this, but whether they happen, when they happen, and how much they happen is different, because the balance of these BCL2 proteins I mentioned at the very beginning defines whether the cell, I mean, they form a very complex interactome, and this defines how sensitive the cell is to trigger apoptosis. So in cancer cells, because they have been exposed to transformation stress, among other, all kinds of stresses, the pro-apoptotic proteins are upregulated, and they are said to be in a state of prime for death, meaning that they are before the precipice, very close to the precipice to decide to die, except that they are kept alive. And that's why the chemotherapy works, because it's actually targeting every cell, but because the cancer cells are closer to trigger apoptosis, they will be killed more effectively. But yes, so a normal cell will have less apoptosis sensitivity. During embryo development, the apoptotic program is very present in the cells, but in adults, for example, some cells do not have high Bax and BAC, so they will be more resistant to apoptosis. You have to imagine, for example, the neurons or the cardiomyocytes, you don't want them to die. So they are very refractory to apoptosis. If apoptosis happens, from all the measurements that have been done in the community, it happens in the same way, but it will not happen because the levels of Bax and BAC are, I mean, you can still kill these cells by apoptosis, but it's more difficult. Answer?
Yeah, there are questions.
You trust me way too much. I have several questions. So first of all, I think this is fascinating. I had never considered this kind of mitotic blabbing is super cool. Now, the way you described it, if I understand correctly, is that you have an active process of the pore formation and pore growth, and then kind of like the way you think about this right now is that, okay, as the pore grows, then you have this extrusion process following that. But I could very well imagine if this is like a physical system that there's feedback, right? So like that the extrusion process itself would also mechanically act on the pore. So kind of like trying to widen it or something like that, so that you have actually like the feedback.
Yes, exactly. This is what we are proposing. We see that the pores with extruded membrane, inner membrane, are larger. In the simulations, we see it.
Okay.
Now we are trying to do more statistics to see, to see that really every time there's extrusion, the pore is larger than without extrusion, and then maybe we can make a correlative.
Okay.
I have to say, we are also playing with the osmotic swelling. We have identified, or in the process of, the protein that permeabilizes the inner membrane. And when we knock it out, then there is less extrusion.
Okay, can you say that again? Like what did you knock out?
So I told you the inner membrane is permeabilized. The mechanism is not defined yet, but we think we have a candidate that mediates it.
Okay.
And when we knock it out, there is less extrusion.
So
it also supports our data that the permeabilization of the inner membrane induces this swelling, and this swelling contributes to the...
Okay, so you really do think this is like an actively driven process, such as osmotic swelling, because it could just be entropic, right? So that you just have there's a polymer, sure, it's going to come out.
Well,
just
from the simulations, also
the energy, right?
The relaxation of the membrane curvature of the crystal contributes, right? So there's also a contribution of.
Okay.
I wouldn't call it. I would say it's a regulated process.
Okay. Fair enough. Super cool. I have more questions, but maybe there's.
Hi, I have a general question about kind of like single molecule tagging, because you mentioned that your bags or bag was actually like 12 kilodaltons long, and then you're tagging it with a fluorescent protein that's also some kilodaltons long. How would this affect the kinetics and dynamics of, like, you showed that like every half an hour there was this change?
So I cannot answer how well as I would want this question, in the sense that if we don't put the tag, we cannot analyze the assembly. But well, with Western blot, but then you don't have kinetics. But what we can tell is that when we have the tag or not, I mean, if you have back double knockout cells, you trigger apoptosis with apoptosis triggers, and they don't die. Now, if you put bags or back with this tag, the cells die. And the process is very similar to when you have bags and back without the tag. This is a very important control that needs to be done. And indeed, it's very important that it is GFP bags and not bags GFP. Because if you have bags GFP, then it doesn't work. So every time we do microscopy, so mostly everything we do in the lab includes labeling. Sorry, guys. But we have to deal with the fact that the protein is labeled, and we need to make controls that the function that we want to investigate is still there. So in this case, the proteins, the tagged proteins rescue the death phenotype very similarly to the untagged proteins.
We have a question online, so I think we can see it here. Okay, Tomo has a question, I think.
So the question is about the...
Yes. What is the order of pore generation from outer to inner membrane?
So we think it's first the outer and then the inner membrane. In a way, the mechanism that... that's an interesting question. Fusion. I mean, now we are starting to do also cryo-ET on the mitochondria, apoptotic mitochondria and cells. So maybe we'll see whether they can fuse. So far, we didn't have the resolution. But what we think it happens based also on this mechanism for inner membrane permeabilization is first the outer membrane is permeabilized and then the inner membrane. Because the protein that permeabilizes the inner membrane is in the cytosol otherwise, so you need a way of it to get access to the inner membrane. Yes, there is also, this is from our experiments, there is also a paper where they measured the outer membrane and inner membrane permeabilization, and they found like 10 minutes difference between the two processes. But when you talk to the authors, to Stephen, he thinks the inner membrane permeabilization timing is not super exact, precise. So, but yeah, that in his hands, the outer membrane is always before the inner membrane.
Thank you, Tom. Yeah. I just have a quick question. I don't know if anybody else has a question, because yeah, might be the last one. You said that you see this blabbing, mitochondrial blabbing for many of them, but is it something that's like, there is a decision of the mitochondria to do this, or is it just during the apoptosis that comes at a certain point and all of them will have it? And in terms of also if it can be rescued or it can be stopped before actually this extrusion?
I don't know.
Okay. No,
the field has for a very long time assumed that the moment the mitochondria are permeabilized is a point of no return in the cell commitment to death. But now it turns out that you can permeabilize mitochondria a little bit. And that is called sublethal mitochondrial permeabilization. And that doesn't lead to cell killing. Okay. So, but however, it's very, very little is understood about this. This is something we want to investigate, and what could be how are bags and back doing sublethal permeabilization, and what is the difference? I mean, what is defining whether it is sublethal or lethal? How is this transition regulated? But I have no answers.
Okay. Okay, if nobody has more questions, really thank Ana again for the great talk. And see you next time.